Okay, let's start with the protoplast fusion or somatic hybridization in a continuation of the tissue lecture of the previous class. In the protoplast fusion or somatic hybridization, what is protoplast? First question is that. So protoplast, if we talk, it is a cell without cell wall, means it is a cell, plant cell in which cell is removed, cell wall is removed. And somatic hybridization means here we are going to take a somatic cells, means non-reproductive cells. Okay, now protoplasts do not have a cell wall. So what it have? It has a nucleoplast, cytoplasm, and cytosol membrane, and this combined is called as a protoplast. So protoplast fusion is a fusion of protoplast of two plants belonging to the different variety. They can be of different variety, different species, and even different genera. Okay, now suppose we are taking a cell A and cell B. In cell A, this is the cell wall and this is the cell membrane of cell A. Then this is the nucleus, this is the vacuole, and this is the other organelle. Same way, this is the cell B. In this, it has a cell wall. Then this is the cell membrane, and then they have a nucleus, and this vacuole, and this is the particular protoplast. And we can say other organelles. Now, both the cells are treated with the enzyme pectinase and cellulase, and this pectinase and cellulase will dissolve the cell wall, so the naked protoplasts are produced. Okay, so this pectinase and cellulase treatment will remove the cell wall. So this is the protoplast of cell A, which has a cell membrane, but this do not have a cell wall. Same way, this is the protoplast of cell B, and here this also have a cell membrane but do not have a cell wall. Now, it is known that this protoplast A is taken and protoplast B is taken, and now these protoplasts are fused in the particularly culture medium, and this protoplast fusion is done by electrofusion, where high frequency of alternating electric current alternating electric current are provided with with short current pulses. Short current pulses. Or the second thing can be done, that is chemofusion. And this chemofusion, if we talk, that chemofusion, that is through the chemical like PEG, that is polyethylene glycol, or sodium nitrate. So either through the electric field or either through the chemicals, these two protoplasts get fused. And when these two protoplasts get fused, this protoplast will look like this. They have two different nucleus. This is nucleus of A protoplast, and this is a nucleus of B protoplast, because here in this protoplast fusion, protoplasm get fused but nucleus cannot fused. And so this cell which is U formed, so hybrid protoplasts are there. So this have a two nucleus. Okay, these have a two nucleus. So we here called this as a what, a particular heterokaryon. Okay, now this heterokaryon, in this the later on we can say what a karyogamy fusion occurs, or we can say the nucleus fusion or karyogamy occur. And when karyogamy occur, the one cell will be obtained with the nucleus of this both is fused. So this is known as somatic hybrid cell, in which the protoplasm, means cytoplasm, and nucleoplasm both are fused. And this hybrid protoplast can be grown further, can be grown further to form a new plant, and these plants are known as these hybrids are known as somatic hybrid, while the process is known as somatic hybridization. So this particular has a fused two nucleus, so this can also be somatic hybrid, can also be called as a synkaryon, because in this the single fused nucleus from this two particular one, and the particularly heterokaryon have a two unfused nucleus. Now the hybrid protoplast is called cytoplasmic hybrid. Now what are cytoplasmic hybrid? So cytoplasmic hybrid or cybrid are there. If we talk about a cytoplasmic hybrid or cybrid, then if in hybrid protoplast, hybrid protoplast, one of the two nucleus, the two nuclei get degenerated, then it is known as cybrid. Suppose this is the heterokaryon, this has a two nucleus. This is A nucleus and this is B nucleus. Now suppose here A nucleus get degenerated, then the further cell will have only B nucleus. Then this is known as cybrid or cytoplasmic hybrid. Okay, so this is the what a cybrid. So this is what a somatic hybridization and the protoplast fusion. Now if we take some particular examples, then we remember that first somatic hybrid, first somatic hybrid if we talk, was obtained by Carlson et al. Carlson et al. And Carlson et al. In 1972. And this first somatic hybrid developed by Carlson et al. Between Nicotiana glauca Nicotiana glauca and Nicotiana Langsdorffi. Okay, so between these two, Nicotiana glauca and Nicotiana Langsdorffi, uh, first somatic hybrid developed by Carlson et al. Okay, there are some intergeneric hybrid, and if we talk about the example of intergeneric somatic hybrid, means here the two different species of two different genus are going to fertilized, or we can say fused. So if we talk about intergeneric hybrid, its example is tomato, and this is a hybrid of potato and tomato. Now we know that potato scientific name is Solanum tuberosum, so potato genus is Solanum, whereas the tomato genus is Lycopersicum. So this is between the two different genus, so it is known as what intergeneric hybrid. Now, this is there, but it is known that unfortunately, Pometo did not have all the desired combination of characteristics for its commercial utilization. Okay, so this is the thing which is seen in case of the particularly we can say the protoplast fusion or somatic hybridization. Now after this, at last we are going to talk about application of plant tissue culture. So if we talk about application of plant tissue culture, the application in, include number one, rapid clonal propagation. And here rapid clonal propagation means by the application of tissue culture, it is possible to achieve propagation of a large number of plants in a very short duration. So in a very short duration of time, large number of plants can be produced. And the method of this producing thousands of plants through tissue culture is called a micropropagation. We remember the method of producing thousands of plants through method of producing thousands of plants through tissue culture tissue culture is called micropropagation. Okay, so through micropropagation, thousands of identical plants will be produced. So how we will do? Suppose we will take one plant, and this from same plant, several test tube are taken, and in this the nutrient media are provided, and in this nutrient medium, the explant from same plant is kept. So this all have a nutrient medium, but along with the explant from the same plant that is here. So this all will produce identical plants because all the explant have a same genotype because they are developed from taken from the same plant. So here all the plants will be identical, all will produce identical plants, not parts, it is plants. And this identical, each of the plant, in these identical plants, each of these plant will have a genetically identical to the parent plant. So they are called, these identical plants are called soma clones because they are developed during tissue culture. And it is known that many important food plants like tomato, banana, apple have been produced on a commercial scale using through this method. So micropropagation means ultimately what? Minute or small cells or small tissues are taken from the parent plant and inoculated in a new nutrient medium in a test tube. The number of test tube you can develop or number of culture medium you can make, and in that you will put a same explant from the same plant. So all will give rise to the identical plants. And after this, the second is pathogen free plants can be produced. Okay, so the recovery of healthy plants from the diseased plant is possible by this meristem culture, and through this pathogen free plant, or we can say through meristem culture resistance to resistance to tongrovirus and leafhopper in rice is developed by tissue culture. Is developed by tissue culture. Okay, so this is the what particularly we can say the pathogen free plants can be produced by this. So these are the applications. Now at last we are going to understand some terminology, and in this terminology we are going to discuss first, that is inbreeding depression. Now inbreeding depression means reduction in fertility or variation or vigor, that is the superiority of offspring over parent, due to continuous selfing, then it is known as inbreeding depression. Now apart from this we need to remember some full name of some organization like NBPGR. So this NBPGR, if we talk, it is a National Bureau of Plant Genetic Resource, Plant Genetic Resource, and is concerned with introduction of germplasm. Apart from this, the next organization, if we talk, that is ICRISAT, that is International Crop Research Institute for semi-arid tropics, concerned with, and this ICRISAT is concerned with storage of germplasm. And the last one is ICAR, and you know that it is a Indian Council of Agricultural Research, and this ICAR is concerned with critical evaluation of newly developed varieties. Of newly developed varieties. So that is what ICRISAT and ICAR. Okay. So this is what about the particularly we can say the tissue culture. Okay. So last take discuss some of the questions. Totipotency means it is a ability of a cell to produce a new plant. Whole plant can be produced from the single cell, then it is known as totipotency. Sterilization of nutrient medium done under the following condition. It is done in the autoclave and it is at 120 degrees Celsius for 15 psi, or we can say pounds per particularly pressure. Now inoculation carried out under condition, that is aseptic condition. Aseptic means sterile condition, that is microfree environment. Okay, so that is all about the tissue culture, and that is all about the plant breeding chapter. I hope you understood well. Thank you.